Hello, everyone. My name is Pawel Szczerbiak, and I'm from the Małopolska Center of Biotechnology at the Jagiellonian University in Krakow, Poland. The title of my talk today is Exploring the Microbiome Protein Structure Space Using Simulations and Deep Learning. I'm sure that all of us at this meeting have their reasons for why do we need protein structures. And this is more true and more relevant now, I think, than in the past years because of all the tremendous progress in protein structure prediction technology. We're able to address more and more questions. The primary goal and the primary question that we are interested in this project is protein function. And our subject of interest is mostly the microbiome. So I've been working in the microbiome area for the past several years. And this is mostly metagenomics, but my structural and molecular biology background is determining and dictating my way of thinking about this issue. So to me, microbiome, the human gut microbiome, or any microbiome for that matter, is essentially a source of genomes. And those genomes encode protein sequences, then protein sequences determine the protein 3D structure, which in turn is responsible for its function. And by understanding the function, we can understand the role which the microbiome plays in health and in disease, and then we can start thinking about designing therapies or designing interventions that would target the microbiome and work for health. But because this is 3D SIG, I'm mostly going to focus on the protein structure and function aspect of the project. So our starting point in this thinking about protein structures and exploring the microbiome is the GABA genomes collection, which is 1003 reference genomes from the microbial part of the tree of life. So this gives us insight into evenly sampled taxa and microbial clades from across the tree of life. This essentially gives us a cross-section of what's happening in the microbial world. From this starting set of genomes, we generated over 2 million raw protein sequences, which we then processed to obtain multiple sequence alignments for putative domains of unknown structure. And using Rosetta and DMP-fold as two complementary structure prediction approaches, we generated de novo models for 250,000 representative domains, which constitute our microbiome immunity project, or MIP for short, structures. Those domains were selected by us to be within the reach of those methods, and that contained enough information for us to build reliable models for them. And using that as a starting point, this is really a great opportunity for us to explore how those complementary approaches of simulation-based Rosetta predictions and deep learning-based DMP-fold predictions, how do they compare against each other? What are some of their inherent biases in the way that they predict protein structures? Of course, as structural bioinformaticians, we're interested in discovering new folds and new protein geometries and protein functions, but this also ties into our willingness to understand structural and biochemical and biophysical features of those proteins and how they can be contextualized in terms of the microbial protein universe. And this is really the last part that we're very much also interested in is how can we contextualize all of this knowledge about microbial proteins and how do they fit into the big picture of all known protein structures? What are some of the um, inherent differences that microbial proteins possess as compared to other types of proteins? And is there anything specific? Is there anything particular about them? In more technical terms, this is what our pipeline looks like. So starting with sequences, we generate, for the same set of sequences, we generate Rosetta and DMP fold models, which we deem to be, for those methods to be, those methods are able to predict, but then we add a layer of model quality assessment to weed out those models that don't fit our criteria, that they don't seem to be too protein-like, and we end up with this set of good models that we then try to annotate and better understand um, what is going on in that space. And because my time today is very limited, I'm going to focus on some overall characteristics of the structures that we annotated. How do they compare between the two complementary approaches of Rosetta and DMP fold? And I'm going to highlight some findings with regards to new folds that we found in this project. Okay, so, but maybe let me just start by telling you what do we consider to be a good model. Well, first of all, we just started with multiple sequence alignments that are large enough to generate high quality constraints, but that is not enough. So for Rosetta, we then consider top 10 lowest energy models and look whether this conformational ensemble that is being generated is consistent internally. And then for DMP fold, we rely on the built-in TM score estimation function that tells us how good the model according to DMP fold is. And then as our proxy for protein likeness, we look at the coil content to try and weed out proteins that are indeed not too protein-like. And having applied all this criteria, we go down for roughly 250,000 models that were originally generated in the project, down to around 200,000 models in our curated data set of high quality models. And I think this is kind of the best summary that I can give you on how those two approaches are different. So looking at cath classification, we focus here on the architecture level, and indeed we can see that quite consistently Rosetta predicts more alpha models, proteins belonging to the alpha class, whereas DMP fold tends to predict more beta and alpha beta models. And this behavior is quite consistent across different architectures. Now, with regards to new folds, we of course had to apply some sort of a working definition. And in terms of our way to approach this topic is we did a structure comparison using TM aligned against cath and against the PDB90 as our points of reference. And then any structural model that is below the cutoff score of TM score of 0.5 against both of those databases is a putative new fold according to us. And this is what this space now looks like as compared to cath. So you can see that the majority of models coming from both methods are indeed quite similar to already known folds and already known structures. But this bottom left lower quadrant is really interesting to us. So this is a space where both DMP fold on the y-axis and Rosetta on the x-axis identify structures as not similar to cath and therefore as putative new folds. How does it look like in terms of numbers? So after we apply both cath and PDB filtering, we're down to 7,500 models for Rosetta and 14,000 models for DMP fold. Um, this is quite a high number, so we wanted to see what it looks like where those two methods actually agree. So for sequences that are the same between the two methods, where both of those methods identify them as putative new folds, we're down to 2,751 models. And then if we additionally apply the criterion of that, not only do we need to predict a new fold, but the new fold according to both of those methods is actually the same new fold, we're down to only 452 models. And looking at this data set, we noticed quite an interesting thing that, in fact, within this set, even though we have proteins that are vastly different in terms of sequence, there are actually some unique structural clusters. And now, depending on the clustering methodology that we use, we end up with between 140 and 158 structural clusters coming from this set of 452 models. So indeed, we can say that this is the area where we are quite confident that we found new folds. And let me just showcase you one of those cases. So here we have a cluster of 87 structures. So this is the largest cluster that we identified, where both Rosetta and DMP fold produces very, very consistent models for vastly different sequences. And indeed, you can appreciate here that the fold is the same and those structures are very similar to one another, with RMSD for corresponding sequences ranging from 0.5 to 2 angstroms. And because very recently we also developed a new function prediction algorithm that takes advantage of both protein sequence and protein structure, we decided to put that to the test in the context of our new folds. So this methodology, which is called DeepFry, uses graph convolutional networks and an LSTM model to embed protein structure with graph convolutional networks and protein sequences with an LSTM to predict protein function. And looking at all of the different models from Rosetta and from DMP fold and all the different sequences in this large cluster, we actually found function predictions to be very consistent between those different sequences, really. So this large cluster that we identified is predicted to be glycosyl transferase and glycosylase activity. Just to show you a little bit more of how we can now combine our structure predictions with DeepFry region-specific function predictions, let me showcase that for the glycosyl transferase activity. So this is class activation mapping. So basically now we can map the function prediction back onto the sequence and structure to look for which residues contribute to DeepFry prediction. So for glycosyl transferase, across all the different structures and sequences within this cluster, we see that for both DMP fold and Rosetta, those predictions are very consistent in this cluster, and we can observe those two large peaks between residues 25 and 41 and 70 and 80. And now we're able to actually map those peaks and the class activation map onto the structure and actually appreciate what region, according to DeepFry, is responsible or contributes to that particular function prediction. So let me just briefly highlight what's next for the project. So within this project, we already predicted 250,000 protein structures, but our ambitions and indeed the size of the microbiome is much, much greater. So within what we call MIP 2.0, we want to predict 1 million protein structures, which I think is definitely within the realm of possibility, given the recent advancements in structure prediction technology. So obviously, such a large project requires a lot of people involved. So this is us from the Małopolska Center for Biotechnology, but our collaborators are from both Flatiron Institute in New York, the University of California, San Diego, and the Broad Institute. This is our team. So basically, this is us in Krakow, me, Pawel, and Mary. Tommy Vatanen from the University of Auckland in New Zealand, and Rich Bonneau's group from the Flatiron Institute in New York. And all of those computations have been made possible thanks to the IBM World Community Grid, and we're very thankful to all the volunteers for their massive computational contributions to the project. And if you want to learn more, please go check out Pawel Szczerbiak's poster, and his poster is a part of the 3D SIG. And if you want to learn more about how we use this knowledge and all those predictions in the context of metagenomes, please go ahead and check out Mary Maranga's poster, which is a part of the microbiome 3D SIG. And I thank you all very much for your attention and welcome any questions. Thanks.